Assalamu alaikum everyone, this is Abdullah Khalil and this is the platform where you can learn O-level, A-level and IGCSE biology of various UK boards. We started biological molecules in the previous lesson. Today we'll start with the emulsion test, the test that is conducted for lipids. We have already seen the structure of lipids. It is made up of glycerol and 3 fatty acid chains. Now we know that lipid is a hydrophobic substance. It cannot dissolve in water, but it can dissolve in ethanol. And that is the property that we use for testing the lipids. In the given sample, we just pour absolute ethanol and we shake it vigorously. If there is any lipid present, it would dissolve in ethanol. And now the second step is to put this ethanol in a beaker of water. Because lipid is insoluble in water, it would come up at the top, forming a cloudy suspension, and that cloudy suspension means that lipid is present in the given sample and the test is positive. Now the next biological molecule that we're going to look at is proteins. Protein is another polymer. Now lipid was not the polymer. Protein is a polymer just like DNA, just like carbohydrates. It is the polymer of amino acids. That means amino acids can join up to make a long chain of polypeptides. A protein may have more than one polypeptides. If you look at the generic structure of amino acids, this is what it looks like. It has a central carbon which is attached to an amino group and a carboxylic acid group. Amino group is here on this side and carboxylic acid group is here. That's why it is called amino acid. Amino acids can join to make peptide linkages to form a very long chain by a simple condensation reaction, as we have seen in carbohydrates and lipids. So a simple condensation reaction would be something like this. Let me draw it for you. So let's imagine this is one amino acid. It has carboxylic acid at the end. This is the carboxylic acid group. And on the other side, we have the amine group or the amino group. The OH from the carboxylic acid and the H from the amino group of another amino acid. They will make a bond forming a water molecule. So this would be removed from here. And this carbon would make a bond with this nitrogen, forming the peptide linkage. This bond is peptide linkage. And this can continue. Another amino acid would come up here with its amine group forming a bond with this OH, and this can continue to form a very long chain of amino acids forming a polypeptide. Now the importance of proteins. Proteins have a very important structural role in living organisms. They are present in the skin, in the nails, in the hair. The enzymes are proteins, the hormones are proteins, most of them, and myoglobin and hemoglobin are proteins. Antibodies are proteins. So let's look at the structure of this protein. First of all, proteins may have 4 different kinds of structures. Primary structure, which is simply amino acids lining up together, making a long chain. That's called the primary structure, which could be represented by this diagram. 124 amino acids lining up, forming the peptide linkages, making a very long chain of amino acids. This is a polypeptide which has a simple primary structure. But if there are hydrogen bondings, that may result in this structure making the folds, causing the folding. That folding could be alpha helix like this, or it could be beta pleated sheets. Alpha helix and beta pleated sheets, both of these are secondary structures. Now let's imagine if we have the primary structure with alpha helix with beta pleated sheets all together forming a specific 3D structure. That's called the tertiary structure. Most of the enzymes have this structure. And up till tertiary structure, we are talking about one polypeptide. If it is more than one polypeptide, it would be called quaternary structure, like we have in hemoglobin. 4 polypeptide chains in hemoglobin. Hemoglobin is a globular protein. Structurally, we have 2 different categories of proteins. We have the globular proteins and we have the fibrous proteins. Globular proteins are so called because their structure is like the globe, spherical. And fibrous proteins, they have the fiber-like, thread-like structure. We will see one example of globular proteins and one example of fibrous proteins. Now, for the globular protein, the prime example is hemoglobin. This is a protein that is present in your red blood cell. It is really important in transporting oxygen to every cell of your body. It has affinity for oxygen. It can make bonds with oxygen. One hemoglobin molecule can make bond with 4 oxygen molecules. It can carry 4 oxygen molecules, and this structure is called the oxyhemoglobin. How does this happen? Now, we have already talked about the polypeptides in hemoglobin. We have 4 polypeptides, 2 alpha and 2 beta. 2 alpha polypeptides and 2 beta polypeptides. And they have been folded into a very specific 3D structure, and the overall structure looks like a sphere. It's a 3D spherical structure. That's why it is called the globular protein. We have 2 beta chains here, and in the center we have a heme group containing the iron. Heme group contains iron atom, and it's the iron atom that basically makes up the bond with oxygen. If somebody has deficiency of hemoglobin, they are recommended the iron supplements, because other than iron, it's just the globular protein which your body can manufacture. But iron is a very strong and it's an essential component of your hemoglobin polypeptides, all the polypeptides. Every polypeptide would have an iron atom which is at the center, which makes the bond with oxygen, which can carry oxygen. And overall, one hemoglobin molecule with its 4 polypeptides can carry 4 oxygen molecules, forming the oxyhemoglobin. Now, this bond is reversible. When the partial pressure of oxygen is high, that means near your lungs, when the blood is near your lungs, near your alveoli, where the partial pressure of oxygen is really high, this reaction will go in the forward direction. That means hemoglobin would react with oxygen to form oxyhemoglobin. But when this is passing through oxygen poor tissues where oxygen is needed, where the partial pressure of oxygen is really low, this reaction would go backwards. So this is important, so the oxygen that is carried from the lungs can easily be transported to the cells. So the bond can easily break down. The types of bonds that are present in proteins, there are 4 different types of bonds. First of all, we have hydrogen bonding. It's the bonding between positive hydrogen and negative end of the molecule. Like you can see here, the positive hydrogen from the amine group and the negative oxygen from the carboxylic acid group can attract each other, and this is an example of hydrogen bonding. Disulfide bond, it's between the sulfur of cysteine with another sulfur of another cysteine. Let's go back and look at the primary structure of protein that we have seen here. If you look closely, you would see that there is an extra bond present between cysteine and cysteine. This is between sulfurs. This cysteine contains a sulfur atom, this cysteine contains a sulfur atom, and they would make a bond together. This is called a disulfide bond. This is the second type of bond present in the proteins. Then we have ionic bonding. Now, the ionic bonding is obviously the attraction between positive ions and the negative ions. We have the positive ions in amino acids and the negative ions which can attract each other, forming the ionic bonds. The last one is hydrophobic interactions. The amino acids may have R group which could be hydrophilic or it could be hydrophobic, making the entire amino acid hydrophilic or hydrophobic. Now, we know that hydrophilic or polar substances may attract each other because of the opposite charges, but we might not know that even for the hydrophobic substances, there is affinity between them. Let me give you an example. Sodium ion cannot pass through the cell membrane. Why? Because the core of the cell membrane is hydrophobic while the sodium ion is positive. So because of being polar, it cannot pass through the hydrophobic core of the cell membrane. It won't be allowed to do that. But estrogen, which is a hormone, can pass through the cell membrane because estrogen itself is also hydrophobic. It is a steroid. So hydrophobic core of the cell membrane has an affinity for hydrophobic estrogen. Similarly, hydrophobic R groups of amino acids have affinity for other hydrophobic R groups. This affinity is called hydrophobic interaction. Now, the last thing here, the structure of collagen. We have seen the structure of globular protein, which was hemoglobin, and now as an example of fibrous protein, we look at the structure of collagen. Collagen is a very strong substance. It makes up your tendons. It is present in your bones. It is present in the walls of your blood vessels. So collagen has a very important structural role, and it is a very strong substance too. Collagen is a triple helix. If you remember the structure of DNA, that is called a double helix, two strands wrapping around each other, twisted around each other. Collagen has a triple helix structure. There are 3 polypeptide chains and forming a triple helix. Now for each turn, there are 3 amino acids present per turn. 3 amino acid per polypeptide per turn. The third amino acid is glycine, which is the smallest of all amino acids. We need the smallest amino acid here. Why? So the turn is very tight. The coiling that is being made here is really tight. Any other amino acid would be too large. The coiling wouldn't be as tight as it is here with glycine, as you can see in the single chain as well. This would form a triple helix, and they have covalent bonds which are holding them together, making the fibrils. Most fibrils would line up together to make the strong collagen fibers. The last thing that we are going to do here is the Bi-uret test, which is the test for proteins. It's a very simple test. You would use Bi-uret reagent. You have a given sample which is to be tested for proteins. You would put a few drops of Bi-uret reagent. Bi-uret reagent is blue in color. Now you would wait for the color change. The magic. If protein is present, the color would change to violet, light purple color. That proves that protein is present. And if the color stays blue, that means there is no protein. Now, as I said in the beginning, I have shared the link of these notes in the description of the video, and there are topical questions available at the end of these notes, so you can attempt all these topical questions and you can refer back if you face any problem. You can write it down in the comments. And if you have learned something today, if this video has been productive for you, make sure to share it with your friends, like the video, and subscribe the channel. Thank you very much, and I'll see you again in the next video.